Recording in progress.
Hi guys, this is the Bio 285 Week 6 review session. Today we're going to talk about pathways, I think. Anything else covered? Just pathways. Cool. You guys see? One thing I was going to mention before we start is we included tips and advice for the class. People were asking me about after the first exam, how to study for this class, how to approach the class. I put slides in the exam one review, but they're also in these slides. So when I email the slideshow later today after the review, you guys can check that out. Thank you. And I hope that the advice helps with moving forward. So now I'm going to pass it over to David to talk about cell signals and receptors. I don't know if you want the microphone, if you want to just
I can just yell, it's fine. All right, guys. So cells receive signals. I tell them what to do. Actually, I'll use it. All right. So is it on? All right. No, still not. Okay. All right. So in human cells, we constantly require signals just to survive. So this is why it's hard to keep human cells alive in labs. Cells receive many signals at once that tell them what to do. So they don't receive just one signal at a time. And these signals are usually made of small ligand or protein molecules. We'll get more on that later. Signals are received by extracellular or intracellular receptors, but in this class, we're going to be talking about mostly about extracellular receptors. Cells receive a number of signals from survival to growth and division to differentiation. And if they do not receive a survival signal, the cell will eventually die, as you can see here. So these are the 4 examples. But also notice how the cells are always receiving their survival signals, even if it's receiving a growth and divide signal and so forth. All right, so to diffuse or not diffuse, right? So signals are typically small molecules or proteins. So hydrophobic molecules or small hydrophobic molecules can pass through the membrane and bind to systolic receptors, example, sex hormones, cortisol, thyroid hormones. And nitric oxide is also hydrophobic. Both the nitrogen and the oxygen are highly electronegative, so they cancel each other out. So like their dipoles will cancel each other out. So we have hydrophilics. They cannot pass through the membrane. They need to be bind to a cell surface receptor. So insulin, growth factors, and signals. So like I said before, we're going to be focusing on cell surface receptors. So over here. All right, so 2 general outcomes of cell signaling. We have a change in protein function, and we also have a change in protein levels in which it's done via gene expression. So both processes are highly regulated. The faster cell response would be to alter existing protein function, and the slower cell response will be to change genes expression. These outcomes are not mutually exclusive, meaning that both of them can result of a signal transduction pathway. So is there any questions so far on that? Yeah.
Wouldn't they get stuck in the
What did she say? The vesicles, right? She mentioned something.
like, but they get stuck within the nonpolar region. If they're nonpolar, they don't want to get critical.
So you talk about this slide. I'm not too sure how to answer that, but for this class, I don't believe you have to know that. I think she just mainly highlights the 2 types of signal groups. So the signals that can pass through the membrane, and those ones are your hydrophobics. And then hydrophilic, if they're hydrophilic, they're polar, they're not going to want to pass through the phospholipid bilayer, which if you remember from class, it has like the lipid tails and the polar heads. And so like the tails kind of face inwards and heads face outwards and kind of form this bilayer. I can show the image at the end of the review, but it's just those 2 groups that you want to focus on.
Yeah, I meant like with the small hydrophobics, like why do they not get through it? They're in like a non-polar. I mean, if you guys can't answer that, maybe it's just like a excited.
I wasn't sure like if inside is polar though, like the cytosol. I don't think she mentioned it in class, but I don't think the cytosol is polar. And if it is, I don't think you need to know that. I'm not sure exactly why the mechanism is that it slides through the membrane, but I think it's just like because the phospholipid membrane has mostly hydrophobic character, that things that are like and like things can just kind of move together. Whereas if you have large signals, or something that's polar, like a different chemical group, then the phospholipid bilayer is not going to be able to pass through. Does that answer your question? Yeah, Jay, did you want to add anything?
No, I'm not 100% sure why. I don't want to say anything wrong, but I don't think Laura ever, I can ask her actually, after
We'll
this,
email her and
just to see.
ask. But yeah, I don't
That's a good
remember.
question though.
Said something about enough to interact with the outside, but not too much.
Yeah, would she say that in class? I feel like I remember her saying that in class. Yeah. Yeah. All right. So same signal, same receptor, different outcomes. So depending on the cell type, the signals may affect different outcomes in the cell. So how? So different cells express different proteins. There are different downstream response proteins for these different receptors. Think of all the different things that happen when you're stressed versus when you're relaxed. Heart rate increases / decreases, digestion speeds up / slows, saliva stops, dry mouth, or accumulates. You want to add something?
Also, I just put the example in here of the parasympathetic nervous system, but you don't need to know it for this class. I just like to link things to the outside world. So yeah.
All right, so one more thing about receptors. So we have extracellular receptors. So signals are usually bulky and hydrophilic. And you have intracellular receptors, and signals are usually small and hydrophobic. This makes sense because they need to be able to pass through the cell membrane. Recall, cell membrane is a phospholipid bilayer, and it's highly selective. The same signal / receptor pairing can produce different outcomes depending on the cell type.
Okay, so now I'm going to talk about signal amplification versus what crosstalk is. And I actually have a video for signal amplification.
Receptor proteins interact.
Sorry, just trying to make it full screen.
With signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
Okay, so that's the video. I thought I'd just put a video in there because it's nice to switch things up to hear things explained and things seen in the video. But that's just a video that explains signal amplification, which I'm going to explain even further. This is full screen. Okay, so I think I'll come back to it. So signal amplification is just like the video was saying, you have one signaling molecule, which is, where's the pointer? Which is pictured at the top, binds to a receptor and can cause the activation of many downstream elements that results in like a large amplified cell response. So just some definitions here. Signal could be amplified if there are enzymes and multiple steps in a transduction cascade. And the outcome is you want to increase concentration of effector proteins in the pathway. And I included like a little biochemistry definition, just the way I learned it in another class, but it's just when you have one signaling molecule, it gets perceived by a receptor, which causes the production of many secondary messengers that go on to bind to many proteins and leads to a transduction cascade. So just having one thing bind to a receptor can cause like an outcome of many things happening. And I'm gonna actually use an analogy, which is why I had that there, to describe what is actually happening. So let's like imagine that like I had like some sort of secret or something that like, let's say the review session, like I told David tomorrow that like I'm going on the Peter Pan bus to go home at like 6:55 a.m. It's not a big secret, but it was all I could come up with. Sorry. So I tell David that I'm going on the Peter Pan bus at like 6:55 a.m. tomorrow on Friday. And then that's just me, one person telling David that. And then let's say David then tells Syed, which is a TA in the class, like the same thing, just sends him a text message. And then like if Syed went and told like 3 other students at his office hours that I was taking the bus at 6:55 a.m. And then each of those 3 students told one other friend in the class that I was taking the bus. Eventually, we would reach somewhere down here, and each of those students told three more of their friends in this class that I was taking the bus. And then, assuming, let's say we had class tomorrow, and you guys all got there, eventually the news would spread fast, and Laura would ask me, why is everybody talking about you going on the bus tomorrow instead of coming to class? So that's just like a random example, which I don't know if that makes sense, but that's basically like thinking of the idea of gossip. Like if you tell one person something, that person tells another person, then the other person tells 3 other people, the news spreads fast. And the effect is that that message is amplified to many people. So that's just the idea with signal amplification. So that's why I have the other diagram. You have the effect of just one signaling molecule, which in this example is epinephrine, causing the activation of 10,000 glycogen phosphorylases, looks like. But that's just like an example of how exponentially you see like signal being amplified in the cell. So does that make sense? If people give me a thumbs up, what I said makes sense. Anyone have questions? Okay, so that's signal amplification. And now I'm going to talk about what crosstalk is, because people tend to get these 2 confused, but they are different. So crosstalk is just when you have components of one pathway affecting another, and a protein from one particular pathway can talk to a different protein in another pathway. So an example in biology is when a cell is undergoing programmed cell death, like if it's receiving some sort of signal for that, then it shouldn't receive a signal for cell division. So one pathway could end up, the pathway for cell division can suppress the cell death like pathway for cell death response. So does that make sense to people? Because I have another analogy for that. But can people give me a thumbs up if that makes sense? Cool. And a big thing to point out is that pathways don't just exist by themselves in the cell. They interact with many different pathways by quote unquote talking to other proteins and different ones. So you can see, excuse me, this diagram up here is the one that we saw in class. You have all of these pathways, survival factor, growth factor, started kind of talking with each other to make things happen in like cell response. So I have another analogy in here. So I'm gonna make this full screen because I realize I actually can't see everything in here. Okay, that's better. Another analogy I have, you have 2 groups of people. You have group A and group B. And so for group A, so let's imagine for these scenarios, you have people in the groups, and the first person has a message that they have to get across the line, the whole line. So the line represents like a group. So you have like group A, group B. The first person has a message. So group A's message is we wear pink on Wednesdays, which you guys know that's from Mean Girls. If you guys ever seen that movie. Then group B, their message is today is Thursday. So the first person in group A is telling the second person that that we wear pink on Wednesdays, excuse me. And then the second group, the first person passes along the message to the second person that today's Thursday. So does that make sense so far? Okay, so then in this analogy, we have group A, and the message was, what was it? We wear pink on, my bad. The message in group A was we wear pink on Wednesdays. And then the message for group B, if you remember, was today's Thursday. Thank you. And let's imagine in group A, the second person gets the message, we wear pink on Wednesdays, and tries to tell the third person, but the third person in group A has fallen asleep. So they're like inactive or deactivated, okay? And then you have group B with their message, today is Thursday, and they're passing their message along just fine, the first person to the second person and onwards in their group. So you can see that the group A, their pathway is disrupted by this person being inactive. So there has to be a way for group A to get their message, we wear pink on Wednesdays, across from person 1 to person 5. So the way that this could possibly work is if you have these two groups, you have group B passing their message like normal, but a person in group B can run up and tap the third person in group A to wake them up. And by waking up the person in group A, you allow group A to successfully pass along their message, which was we wear pink on Wednesday. And then group B can pass along their message, today is Thursday. So does that make sense to people? Just like an analogy, instead of using science, I use groups and messages. Can people give me a thumbs up if that makes sense? Sounded better in my head, I feel like. But anyways, so you see in the analogy I said, you have two groups, group A, group B. The people represent proteins, and then the two groups are two different pathways. And the message that was being sent for each group can represent a signal being transmitted across a pathway. And the concept that we're trying to get at here is crosstalk. So all those things you can see in the analogy I drew out. It took me a while to draw that out. I was kind of proud. Okay. All right, now we're gonna move on to talk about RTKs. So RTK stands for receptor tyrosine kinase. So to be able to talk about RTKs, I just threw in a little something in there to understand what kinases are. So a kinase, if you guys recall from either 151 or I don't know if we talked about it at the beginning of class, did we? Yeah, maybe a little, maybe a tiny bit. But kinases are proteins and they're examples of enzymes as well. And there's a wide range of kinases. Don't think you have to know like the different categories of kinases there. I just put it in there. So kinases, what they do is they're enzymes, and their enzymatic reaction is to cleave phosphates from ATP molecules and stick them onto other substrates. And other substrates in this class usually mean a protein. So kinases phosphorylate things. So when they attach phosphates to proteins, they phosphorylate them. I have two diagrams here, so you can see the kinase phosphorylating the protein. So I put this in here just for you guys to understand that kinases don't just stick yellow peas on things, because you'll see that a lot in your diagrams, but really behind the scenes, there's a more biological mechanism happening. So you guys can understand that they don't just stick peas on things, they will cleave off phosphates from ATP and stick them onto substrate. So that's just like something for you guys to understand what is happening like mechanistically in the pathways. And yeah, another diagram. Any questions on that part so far? Okay, so now we know what kinases are. So we can talk about what RTKs are. So RTKs essentially are receptor tyrosine kinases, and they are composed of three parts. So the first part we'll talk about is the part that actually binds to the signal. So it has a receptor domain, and then it also has the part that's, embedded in the receptor, not in the receptor, sorry, long day, embedded in the like membrane a little bit, which is the transmembrane domain. And then the third part, which is on the inside of the cell, is your intracellular portion, and that portion has the kinase domain, or the tyrosine kinase domain. And yeah, so those are the three parts of an RTK. And RTKs, essentially, they're just families of receptors used for a wide range of things like cell division, differentiation, insulin signaling. And yeah, so that's what an RTK is. And now I'm going to talk about what it does. Anyone have any questions so far? People give me like head nods, thumbs up. I'm making sense. Thank you. Okay, so now I'm going to talk about how RTKs work so that we can go on to talk about how these pathways work. So generally what happens, you have RTK, not RTK, excuse me. You have the signal will go and bind to the receptor, the RTK, the top portion of it. And when the signal binds to the receptor, as shown here, the two RTK monomers, which are the two like blue pieces, they will dimerize, which is just a fancy way of saying that they come together. They come close in proximity to each other. And when the RTK is dimerized, you get the kinase activity. It says kinase activity stimulated there, which just means the intracellular kinase domains become active. And then when this happens, you have cross-phosphorylation of the interest in the RTK, specifically in the intracellular portion. So on the tyrosine kinase domain, so they come together, they dimerize, and they will cross-phosphorylate each other. So each little RTK monomer will phosphorylate the opposite one. So that's the cross-phosphorylation, or also called autophosphorylation. And yeah, so once they will dimerize, cross-phosphorylate each other, they now become active RTKs. And an activated RTK is able to create binding sites for proteins involved in these pathways. Any questions so far on anything I just said? Yep, you got a question.
What's the function
Sorry, can you repeat that one time?
what's the function of the tyrosine kinase domain? Is it just to phosphorylate the
For the purposes of this class, I believe that's what you have to know. Yeah.
Yes.
Yeah, so for the phosphorylation, the autophosphorylation occurring in the RTKs, it's just like within the tyrosines located on the tyrosine kinase domains on the inside portion of the RTK. So that's what's going on here. That's like their main purpose. Yeah, that's all I can think of. I think. That's should be what you have to know for this class for that. And oh, another thing I forgot to say is your signaling molecule has to always be in the form of a dimer for the RTK to dimerize and do its function normally. Okay. And I included this slide that just talks about details of RTK activation. I think I put it in a little bit like simpler terms-ish on the other slide, but I'm sending you all these slides by email at the end. But yeah, I don't feel the need to repeat myself unless anyone wants me to. Does that make sense? Okay, now we're gonna jump into the actual pathways that we spoke about this week. There are 4. We got RTK, RAS, MAPK, survival, JAK-STAT, and GPCR. All these pathways have their differences, but they also have similarities. And they're all eukaryotic pathways, I believe. For the JAK-STAT, the example that we discussed in class, it uses like milk protein production, which is done with prolactin, I believe, for breastfeeding. So that's the pathway we use when we talk about JAK-STAT. And yeah, so we're gonna continue to talk about these. And I'll just hand this to David.
All right, so the first pathway that Laura went over in class was the RTK RAS MAPK pathway. So sometimes referred to as the growth factor signaling or as RAS RAF MECRC pathway, but you don't need to know that. Okay, so let's see. So basically, like Michelle was talking about, how the pathway starts is when we have a signal molecule in the form of a dimer is going to bind to the inactive RTK on the signal on the receptor domain, right? And then once the signal binds to the tyrosine kinase domain, or not the tyrosine, once the signal binds to the receptor domain, the RTK is going to dimerize, right? And this dimerization is going to cause the cross phosphorylation on the tyrosine kinase domain, right? So think of it as on the tyrosine amino acid, the phosphate is getting added to that, and the kinase is what's doing the hydrolyzation for that. So, all right. So this is kind of like the whole pathway. So we just talked about the beginning part. So any questions on that first part? All good for there? All right. So after that occurs, a protein in this diagram, it says adapter protein, but I think Laura just talked about GRAB2. So GRAB2 is going to bind to this phosphate through the SH2 domain, which is right here on GRAB2. right? So it's going to adaptive protein binds to the phosphorylated tyrosine. It's going to recruit a RAS activating protein, which is a RAS-GEF, aka SOS. I think Laura also talked about that. And I don't know if Laura said this, but this is an SH3 domain on the GRAB2, and then this is the PXP motif on the RAS activating protein. Pardon?
Okay, so for this one, the adaptive protein can also be called GRAF2. You can use either term, but there are two domains pictured here. So I believe for on the activated RTK and where the activated RTK connect and where the adaptive protein, where David is, can you go over to that? Yeah, so over there, that's SH2 domain, the phosphorylated tyrosine area. And then the SH3 domain, which can't be like seen there, but I believe that's what connects the adaptive protein and the RAS activating protein, which can also be called RAS-GEF, I believe, the RAS activating proteins, the lighter blue.
All right. So once RAS activating protein binds, it's going to swap. It's going to cause inactive RAS to swap GDP for GTP. And now RAS is now active due to the conformational change. So phosphotyrosine binding domain, SH2 domain. And also keep in mind that RAS is anchored by a cell membrane lipid. All right. So now we have active RAS. And now RAS is going to activate MAPK3, but keep in mind that RAS is not a kinase, so it's not going to be phosphorylating MAPK3. That's like a really important thing that Laura will definitely test you on. So activated RAS interacts with MAPK3, and MAPK3 is going to phosphorylate MAPK2 at two different positions. The MAPK2 is going to phosphorylate MAPK1 at two different positions, and then MAPK1 is going to phosphorylate other proteins. Just a quick reminder, remember that RAS is not a kinase. It is not known how it activates MAPK3. In other words, RAS does not phosphorylate MAP kinase, or MAPK3.
Just thought I'd point it out if it ever comes up as like a question on the exam and you had to explain like how the pathway worked. You can at first you can write out like MAP kinase, but that might be a little repetitive to write out each time. So you can write out like MAP kinase, and then abbreviation MAPK3. And then from there on out, you can do like MAPK3 and abbreviate to save your hand from cramping when you're writing stuff. And yeah, I thought I'd include that. And then the MAP kinase stuff is just like what we call a kinase cascade. Okay, any questions on that?
All right, so this is kind of just a summary slide of what I just talked about. So this would be a really great slide to study off of. All right, do you want to do that one?
And then David and I just included at the end of each pathway, there's a slide that's just an empty slide with a photo of the pathway. And so you guys can, when you guys are studying or looking at these pathways, you can go back and kind of test yourself and see, like, can I tell what's going on here? So you could just say to yourself, okay, so the signal binds to the RTK dimerizes, they cross-phosphorylate each other. And then the RTK monomers cross-phosphorylate each other. And then the RTKs are active. And then adaptive protein binds to the active RTK on the SH2 domain and blah, blah, blah. So you guys can go through and practice out loud saying what's going on in the pathway so you can kind of test yourself. So each of the pathways that we describe are broken down into small chunks, so it's not overwhelming to see. But then at the end, we include the entire diagram so you can put it together. Does that make sense? Okay, now the second of the 4 pathways, survival pathway. So just key things to think about for a survival pathway. What's the end result of this pathway? What's the ultimate outcome? And can you describe what happens in the pathway to someone who's never taken this class before? And another thing I thought about, which is mostly the types of questions you'll see on like problem set, possibility on the exams, because we know Laura loves to give us problem solving questions, is how could you cause the cells to die in the survival signaling pathway? Okay, next slide. Go ahead. Sorry, I didn't know this slide was animated. Anyway, so what does survival pathway do? Survival pathway works to promote survival, which in reverse terms would mean inhibit programmed cell death. So cells are typically programmed, their default setting. So like just think of like your phone, like the default setting for like the language is English or something. But like a cell's default setting, excuse me, is cell apoptosis, which means cell death. So what the survival pathway does is it inhibits cell death and helps the cell to survive. And something I remember Laura told me when we were studying this pathway, it's important to researchers and biologists. And cancer biology research often targets this pathway because, as you know, cancer cells divide, and replicate. And so you want to stop them from proliferating. So that's why it's interesting to study this pathway. And this pathway will work in harmony with other pathways to help the cell survive. So it doesn't just work on its own, which we talked about earlier, the crosstalk. Okay, so beginning of this pathway, which shouldn't surprise you, beginning of most pathways we talk about is you have the signal binds to the receptor. So the signal will bind to the receptor on the RTK, and signal binding to the receptor will cause the RTK monomers to dimerize, cross-phosphorylate each other. And so the RTK becomes active. Once the RTK becomes active, that creates a SH2 domain on the phosphorylated tyrosine area. I kind of like to just point to stuff. That creates binding site for PI3 kinase to bind to. And so I think PI3 kinase, what it stands for is that one word, I can't even, phosphatidylene. Yeah, no, it's fine, I won't say it. But Laura says it in one of her lectures, so I'll let her pronounce that. But you have, anyways, after RTK becomes active, the PI3 kinase will bind on the SH2 domain on the active RTK. So it will bind to that spot. And then PI3 kinase will phosphorylate inositol phospholipid, which is, which is pictured above. So inositol phospholipid is not a protein, hence the name phospholipid. So inositol phospholipid starts off as diphosphorylated when it's like just on its own minding its business in the cell. And so when it gets activated by PI3 kinase, that'll cause the inositol phospholipid to become triphosphorylated. So in its triphosphorylated state, which we just call phosphorylated inositol phospholipid, it is now active to do things. And so you see that pictured there. Any questions so far? Yeah, and also in its phosphorylated state, inositol phospholipid can bind to protein kinase 1. Any questions so far? No? Thumbs up if it makes sense. Thank you. I rely on the approval. So thank you too. Anyways, so you have phosphorylated inositol phospholipid. It's a mouthful. Once you have phosphorylated inositol phospholipid, it's going to be able to bind to AKT, which is the green protein pictured there. Now you have to imagine, because this is a cell, we don't just have one phosphorylated inositol phospholipid, we have multiple just around the cell. So you can see phosphorylated inositol phospholipid will bind to protein kinase 1 and AKT because it has the binding sites for it. You can tell on the shape because it can sit the triphosphorylated inositol phospholipid, which I think can be abbreviated to PIP. So anyways, and because PIP, which is phosphorylated inositol phospholipid, can bind to AKT, it can bind there because AKT has a pH domain. So that's just another one of the domains Laura talks about. There are 3. So there's SH2, SH3, and I think pH domain. So AKT, which is the green protein, has to be phosphorylated by two kinases to be activated. So when PIP, which is the phosphorylated inositol phospholipid, sits on the AKT protein, AKT is not active at that point because it has to be phosphorylated by protein kinase 1, PK1, and PK2. And so this happens subsequently. How to say it? Subsequently? I don't know why I struggle with that. Anyways, so protein kinase 1 will phosphorylate AKT on one side. And when that happens, it causes a conformational shift in AKT, which allows for protein kinase 2 to bind to AKT on the other side, which is pictured there. So PK1 has to go first before PK2 in that phosphorylation sequence. And protein kinase 1, which I mentioned before, also can bind to PIP3 because it has a pH domain. And so once AKT becomes activated, so remember for AKT to become activated, and it has to have the PIP3 binding in the pH domain, and then also PK1 phosphorylating on one side and PK2 phosphorylating the other side. So then those are all the things or ingredients required to make AKT active. Then when AKT is active, it will dissociate from an inositol phospholipid and enter the cytosol, and dissociates from the other things too. So now we're getting towards the end of the survival pathway. So what happens here, we have activated AKT at this point, dissociates from an inositol phospholipid, enters the cytosol, just said that. So then when that happens, you have AKT, which is the green protein, will bind to BAD. And BAD is the blue one in the diagram. So AKT will bind to BAD, and active AKT will phosphorylate BAD, which inactivates BAD. And that's not a typo, actually. That actually happens. In this class, you'll probably be used to seeing phosphorylation activating things, but phosphorylation can deactivate things also. So big P here, when AKT phosphorylates BAD, it inactivates it. So what happens is, before AKT phosphorylates BAD, BAD at that point is active. And when BAD is active, it is bound to the red protein called BCL2. And so BCL2 essentially is what we call apoptosis inhibitory protein. And an apoptosis inhibitory protein prevents cell death. And so when before BAD is phosphorylated, it's in its active state and it's bound to BCL2. And if it's bound to the apoptosis inhibitory protein, then that means it would be causing cell death to happen, which can be a little bit confusing, but I'm going to get to it on the next few slides. So anyways, we have active AKT. It phosphorylates BAD, which causes BAD to become inactive. And when BAD is in its inactive state, it'll cause BAD to dissociate from BCL2 and bind to this gray protein in the corner over there. You don't have to know the specific name of that protein, but just know that when BAD is inactive, when BAD becomes phosphorylated by AKT, it becomes inactive and it binds to a different protein that is not BCL2. And so when that happens, BAD becomes inactive, it's no longer bound to BCL2. And when BCL2 is free, it's actively preventing cell death. So apoptosis inhibitory protein. And when it becomes active, it's inhibiting cell death. Any questions on that so far? I'm going to get into it a little bit more in the next few slides because I know it can be a bit confusing, that part. Next. Okay, so a little bit more about how that works. So apoptosis, cell death. BAD is a protein that causes apoptosis to happen, and BCL2 is a protein that inhibits apoptosis. So when you have active BAD that's bound to BCL2, BCL2 is at that point inactive, or it's blocked by BAD, and that can cause apoptosis, which in the cell is indicated by mitochondrial pulse. And so that's one scenario. But then in another scenario, when we have the survival signaling working properly, you have inactive BAD, which, if you remember, happens when active AKT phosphorylates BAD, causes BAD to become inactive. When BAD is inactive, it can no longer bind to BCL2. And so if BAD is not bound to BCL2, then that means BCL2 is active because it's not held in captivity by BAD, and it can inhibit apoptosis, which means inhibiting cell death. So in other words, cell survival. So does that make sense to people? Can I get a thumbs up? Because I know that part's like a little bit tricky. Okay, next slide. So I have those scenarios just pictured there. So you have BAD bound to BCL2, And so the inhibitory protein is inhibited. So if you inhibit the inhibitor, so it's like a double negative, you get cell death. And then the other scenario, which is what I said, you have active AKT, which will phosphorylate VAD, causes VAD to remove itself from BCL2 and bind to a different protein. And BCL2 is no longer being held in captivity, so it's free to go and do its job. And what is its job? Its job is to prevent cell death from happening. So then cell death does not happen and you get cell survival because the apoptosis inhibitory protein is active, so it's actively inhibiting cell death, so cell survival. Does that make sense? I just try to picture it in different ways because I know it's difficult to understand that part. Any questions? No? Okay, so this slide here is just a summary of all the steps of the survival signaling pathway, something useful to study, and just in a different form than the pictures. Next slide. And like I said earlier, practice slide to practice the pathway, figure out what's going on normally, so that you guys are prepared to answer questions that involve like mutations and whatnot, or what could happen that could cause cells to die or something. Okay, next slide.
All right, so the JAK-STAT pathway. Just a couple questions. Is JAK-STAT considered to be an RTK, receptor tyrosine kinase? How does the JAK-STAT pathway work? Can you explain all the steps in this pathway to someone who's never taken Laura's Bio 285 class before? All right, so let's get started here. So quick recap on what receptor tyrosine kinases are. So RTKs are monomers with extracellular receptor binding domains and intracellular tyrosine kinase domains. So kinase, remember, is an enzyme that catalyzes phosphorylation. And in this class, most of the enzymes will deal with our proteins. How RTKs work. So a signaling molecule dimer causes dimerization of the RTK monomers whose tyrosine kinase domains interact to stimulate kinase activity. This interaction causes autophosphorylation of tyrosines on the intracellular portion of the RTK, which creates binding sites for intracellular proteins, like we talked about before. Okay, so let's get into the JAK-STAT pathway. So this pathway is involved in many processes, so immune development, mammary gland development, lactation, adipogenesis, but we're going to mainly just talk about the prolactin with milk protein genes. So remember that JAK is a receptor, it's not an RTK. It is a receptor, but does not have a kinase domain. So instead it has kinase subunits that associate with it. JAK is not a receptor tyrosine kinase, just to reiterate, because a lot of people might get that wrong on the exam. This is because it is not a receptor monomer that cross-phosphorylates each other, but instead is the JAK protein themselves that do the cross-phosphorylation in this pathway. The cross-phosphorylation of each JAK protein is what causes them to become active. These JAK proteins just happen to be attached to the receptor. And as we said before, that the receptors are not RTKs. And something that Laura asked, so JAK literally just stands for just another kinase. So it recently took on the new name Janus kinase. Yeah, but so remember that it's a type of non-receptor tyrosine kinase. JAK is not a type of RTK.
Let me see. I just want to re-explain that. I know David said like 3 times, but it's not an RTK, and that's because this the receptors that are involved in the JAK-STAT pathway, they themselves don't intrinsically have like kinase activity. The receptors are just receptors, and then they just have like proteins. The blue objects are just clipped on or just attached to them. So the proteins themselves are what have the kinase activity, and they're just tacked onto the receptor. So that's just key to know. Because RTKs, like their proteins and their receptors, like built into the structure itself. But for the JAK-STAT pathway, you just have these prolactin receptors, and then they have proteins tacked onto them. So just the key thing. Sorry.
All right. So now let's go into what I believe is the actual pathway. So, so very similar. So we're going to be talking about prolactin. So prolactin. Oh, it's fine. So prolactin is going to bind to the prolactin receptors, and that's going to cause the receptor in the pathway to dimerize. Right now, once it's dimerized, each of the JAK proteins, so we have JAK1 and JAK2, they're going to phosphorylate so we have JAK1, so JAK1 is going to phosphorylate JAK2, and JAK2 is going to phosphorylate JAK1. So like cross phosphorylation, like we talked about before. So once that happens, each JAK is going to phosphorylate its own receptor. So JAK1 phosphorylates here, JAK2 phosphorylates here, right? So once that happens, that's going to cause a protein called STAT. And there's a lot of types of STAT proteins, but we'll just be talking about STAT5 in this pathway. That's going to cause STAT5 to bind via the SH2 domain to the phosphorylated receptor, right? Now, once that happens, JAK1 and JAK2 are then going to phosphorylate STAT in this position. Remember, they're phosphorylating the JAK or the STAT on their side of the receptor, right? So once that happens, conformational change is going to cause STAT to disassociate from the receptor. STAT's going to form a dimer. This is another SH2 domain binding on the phosphorotyrosine sites. And it's going to enter the nucleus and act as a transcription factor. And then to relate back to what we've already learned, this is done via importin. We did nuclear import and export. Any questions on that? Yeah.
So I'm just a little confused again about the JAK not being kinase. If it doesn't have any kinase activity, how is it able to like phosphorylate and then phosphorylate others?
Okay, so the JAK, maybe we confused you a bit there, but the JAK proteins, I believe the blue proteins, they are kinases. But what we were trying to refer to earlier is that the JAK receptors, which are the green receptors, those are not what we call RTKs. Because if you remember, RTK, receptor tyrosine kinase, like all that kinase activity and phosphorylation stuff is built into the intracellular portion of this receptor. And so that's different because the receptors can autophosphorylate each other and then cause proteins to bind on the SH2 domains on the receptor. Whereas for the JAK-STAT pathway, it's different because the JAK proteins themselves, which are just clipped onto the sides of the receptor, these are involved in cross-phosphorylating each other. And then they have to phosphorylate the receptor first for the receptor to be able to bind to other things. Because the green receptor themselves do not have kinase activity. It's the JAK proteins, which are the blue parts, that are the kinases in this pathway. So does that make sense?
Yeah, so if someone just says JAK, are they referring to the protein or are they referring to like the receptor?
I'd say protein.
The protein?
Yeah, I don't
I'd say
know.
protein,
I would
but-
say protein for that one.
Yeah. I get what you mean though, because for the pathway, the name JAK-STAT can be kind of like confusing. But if she talks to you about the JAKs, like I think she would say JAK1, JAK2 proteins or something, she would specify. But if there's ever a time where you really don't know, like in an exam or something, you're always welcome to ask one of us or Laura and she will clarify to you, I mean the protein. Like that's a fair question to ask.
Any other questions on that? Good.
Oh, wait. Sorry, I'm always like interrupting here. But yeah, in this pathway, how the STAT5 dimer travels to the nucleus, right, which is this like gray area where it's going to, it gets there with importance help. It's not pictured there, but it's just something cool to know. Links everything.
And then like before, this is just like a summary slide of what I just talked about. And the practice slide for you guys before the exam. All right, so the last pathway Laura talked about was G protein coupled receptor, GPCR. So just some questions. So how does GPCR signaling work? So are you going to be able to explain the steps, this pathway to someone who's never taken this class before? So we'll answer these questions in the next slide, but what is a GAF analog in the GPCR signaling and what is a GAP analog in the GPCR signaling? Because Laura wants you to connect all these concepts together. So this is just an example diagram, but we'll run through it on the next page if you want. Actually, maybe we can just do it on this page and then I'll check to see if I missed anything. So remember the GPCR is a 7 transmembrane receptor. So we have our extracellular receptor binding domain, and we have intracellular components. So we have our G protein, which is made of G alpha, G beta, and G gamma. Just know that the only protein or the only G proteins in here that have a phospholipid are G gamma and G alpha. And also keep in mind during this entire pathway, G beta and G gamma always stay together. So basically how it would work is, oh, also one more thing I want to add is that G alpha is a GTPase. So it can hydrolyze GDP for GTP, which we'll talk about later, and vice versa. All right.
No, GTPase just does GTP to GDP.
Yeah.
Not vice versa.
Just GDP to GTP. That's yeah. All right. So the first thing that's going to happen is we're going to have a molecule come and bind to the receptor, right? And once this molecule binds to the receptor, it's going to cause G alpha to lose its affinity for GDP, and it's going to cause GTP to come in. And that's going to cause G alpha and G beta gamma to separate from each other. So when they separate, they both can have their own effector function. So G beta and G gamma are always going to stay together. They're going to diffuse, I guess, horizontally across the cell's membrane, and they can have an effector function within the cell. And then also G alpha can have a function within the cell at the same time. So when GPCR proteins get ready to turn off, we're going to be using another protein called regulator of G protein signaling, which is an RGS right here. And RGS is going to bind to G alpha, and it's going to cause the G alpha to lose its affinity for GTP to in exchange have GDP diffuse in. And then once GDP diffuses in, the receptor is going to be in its off conformation again.
Yeah. I was just gonna say for like purposes of this class, I don't know if you guys studied this pathway in other classes like biochem or something, but for this pathway, Laura mostly focuses on like G alpha, mostly what it does, like beta and gamma, like beyond this point, she doesn't really focus on. So once G alpha is bound to GTP, it dissociates and does its own thing with signaling. But yeah, the main focus in this part, she tends to focus on just G alpha in this area. So yeah, when you're explaining, you can explain that G alpha just dissociates from GPCR, and you can just say like beta and gamma, like David said, stay together. But just the focus tends to be like in the splitting up part is more about like G alpha doing its thing, and then it comes back with the pathway when GPCR gets deactivated.
Also, just to mention that RGS can cause G alpha to lose affinity for GTP or GTP to pick up GDP. But also, I believe Laura said that G alpha and its downstream effector function can run into another molecule that can cause G alpha to lose affinity for GTP and pick up GDP. I think she mentioned that. Yeah, hydrolyze. All right. So this kind of just talks about what I just said, but for the analog, which Laura will 100% probably ask you on the exam, the GPCR is the GEF analog, and the RGS is the GAP analog within that pathway. So any questions on GPCR? Yeah, so does anyone want to explain why they're analog since we've learned GEF and GAP before? Does anyone? Go for it. Yeah. Anyone? Yeah, go for it.
So GPCR is the GAP analog because when the receptor like gets bound to by a signal, then G alpha will exchange GDP for GTP through dissociation and like confirmation change, whatever. And then so that's like turning it on like a RAS gap and the other stuff we learned. And then RGS will come and then change the affinity of RAS for GTP, so it dissociates GTP and then GDP binds, so it's like RAS gap, it's turning it on.
Good response.
You wanna like
Yeah, I'm just gonna
reiterate
like
for the
repeat it in the mic just so the people
who's
on Zoom can hear
I
it.
should have just given you the mic.
So
TA.
the GPCR GEF analog, because we're going from GDP to GTP, and RGS is the GAP analog because we're going from GTP to GDP. Twice.
And yeah, I'm just gonna add something because I always like to add things. But GEF basically helps to activate I'm sorry, excuse me, what am I saying? GPCR acts like the GEF analog or behaves like GEF because GPCR helps to activate G alpha. And then you have RGS, which kind of behaves like GAP in a way. That's why we say RGS is GAP analog, because it's helping in the process of like deactivating the G alpha. Because remember, G alpha is an enzyme and it specifically GTPase, so it has its own ability to hydrolyze GTP and GDP, so it can like inactivate itself. But RGS just can bind to G alpha and accelerate the hydrolysis of it. So same way that GAP does not physically hydrolyze, RAN in the nuclear import export, it just like helps speed up the hydrolysis process of RAN. But yeah, just applying what you learned before to the stuff. Just an image of.
And this is just an image about what we talked about. Maybe it's a little bit better than the other image. So the other image looks like it was made in the 90s. And then that's the other image if you guys want to use that one too for studying. And wrap up when we should.
Okay. Yeah, thanks you guys for moving along with us. We're almost done here. So just thought I'd include, oh, that came out really tiny. I'm just realizing that. Sorry, I'm going to make it larger later. So just talking about now ways to shut down a pathway, because we know that Laura or Professor Francis loves to ask us questions about what can happen when we take something normal, make it abnormal. But basically, this is about shutting down pathways. So to be able to shut down a pathway, to cause it to shut down and have a large effect, you have to inhibit the effector, the last effector, last component of the pathway that's been activated. And you can shut down a pathway for sure, like 110% you're sure, because you're preventing the end goal from happening. And so in pathways, we know that we have signal binds, causes a bunch of things to become phosphorylated or kinase cascade. Then at the end, you get cell response. And so if you cut off cell response from happening or stop that from happening, you've successfully shut down a pathway. And so cell response essentially happens when you have like signal being transduced, and then it makes its way to the nucleus, and you activate transcription factors and you get cell response. And so that's when like the last component of the pathway, like the last protein, contributes to activating a transcription factor, or the last component could be a transcription factor that enters the nucleus and leads to cell response. So example I have is cell survival happening in the survival pathway. But if you somehow inactivated the last component of that pathway, you could possibly stop cell survival from happening. So you could get like cell death from happening. But an example of like an end goal happening is when you have gain of function mutation, so causing BCL2 to be active even if there's no signal bound. So you'll get cell survival then. So you'll have the cell response happening. But there are ways that you can deactivate a signal in a pathway, which may or may not permanently shut a pathway down. And that's because you could potentially trigger the last component of a pathway to be activated through crosstalk or some other method. But obviously, this sounds very vague, but depends on what scenario is given. So if you had in the RTK RAS pathway, MAPK pathway thing, if you had the signaling molecule gets degraded, and then you had a mutation in MAP kinase 1 that caused it to be constitutively active, which means that it was always active even if you had signal or no signal, then MAPK1, which is the last component in that pathway, can enter the nucleus and can cause cell response to happen. So you have cell survival. Even though you've tossed away the signal molecule, you can still get not cell survival, excuse me. It's the growth factor pathway, so you get cell growth. So you'd have the cell response still happening, even though you took away the signal. So does that make sense to people? So just thinking about ways you could shut down the pathway and ways you can like keep the pathway always on or something like that. Just cool, interesting things to think about when it comes to answering problems like that. Another way you can like deactivate signal or shut down a pathway is if you had receptor-mediated endocytosis. So if you had receptor-mediated endocytosis, which just means the cell like swallows the receptor or it gets engulfed in the cell, there's nowhere for the signal to bind, right? And most of these pathways start with a signal. I think all of them do. So if you get rid of the signal of a pathway, nothing else happens. The signal can't bind to the RTK, no dimerization, no autophosphorylation, no autophosphorylation. You can't activate the downstream effectors. So then the pathway just doesn't happen. So for survival pathway, that would result in cell death, obviously, which is the cell's default mechanism. So those are just things to think about when we're trying to shut down a pathway and thinking about how pathway could be always on. Any questions about that? Just wanted to point out like common misconceptions. You guys already know about GAPs and GAPs, so I'm not going to go into much detail about it unless anyone wants me to at the end. But remember, GAP does not hydrolyze stuff, it just accelerates the hydrolysis of a particular enzyme or GTPase. And GAP will promote GTP binding into a binding pocket of a certain protein. So you can change the affinity of a protein so that it has weaker affinity for GDP, stronger affinity for GTP. When GF binds to a protein, causes a conformational shift in that protein. And so it can no longer hold GDP, so it will want to bind a GTP. So it can drop GDP and exchange it. So that's GF exchange, like guanosine nucleotide exchange factor. That's what GF stands for. And example of enzymes, when we're looking at these pathways, not all proteins are enzymes. So it just depends on what a particular protein's function is, whether it would be classified as an enzyme. So things that can intrinsically hydrolyze stuff, like G-alpha and GPCR pathway, or kinases are examples of enzymes, so the JAK proteins. So not every single protein that you see in the pathway can be an enzyme. You just have to keep in mind what exactly is that protein doing, and you'll be able to tell. And pathways are always highly regulated. If you always have a pathway always on, just like keep in mind, like it's not good for a pathway to be overly active, so having too much signal and even too little signal. And just something to keep in mind, like if you're explaining a pathway, it's always good to make sure if the question asks you to explain like a particular pathway, to just explain like everything that's going on and like functions of stuff, proteins and functions when it comes to like a particular question. If you're not sure, you can always ask too. Any questions? Okay, and that's it for the review session. Thanks for powering through. These videos can be long on YouTube, so thank you for people watching on YouTube as well. And we all the TAs encourage you guys to view the wordless slides, so the slides with just the image, practice how these pathways work, talk to your parents, friends who are not in this class, your goldfish. I know people have a goldfish pet, but whatever, just try to say these pathways out loud, because I feel like you'll be able to catch gaps in your knowledge, or if you're group studying, to be able to understand what's going on here. And if you can accurately explain what happens to somebody else, then that shows that you've understood the concept. And key idea for pathways, basically understanding what happens in a pathway in normal circumstances, so you're prepared to answer questions for when things get switched up. And yeah, so that's it for the review session. And that's pretty much it.